Hello, everyone. As I said, my name is Rodney Alone. I appreciate everyone coming out to the talk today. We had so many people that we actually have to videotape it for those who can't attend, so it's great to see such a big turnout. The work I'm going to be talking about today is the capsules for object segmentation. This is joint work I did with my research advisor, Dr. Ulas Bagci, at the University of Central Florida at the Center for Research in Computer Vision. This talk is going to consist of three main parts, which is sort of why should we use capsule networks? What the heck are capsule networks? And where do we go with these things? I'll talk about the novel extensions that we proposed in this paper to the capsule network architecture. And then we'll get into sort of the medical application we chose to apply this to and the results of that application. So why capsules? Just out of curiosity, how many people in here have at least heard of Geoffrey Hinton's new capsule network architecture? Almost everybody. Wow, that's very exciting. That was unexpected. Awesome. So maybe you guys are already aware of this. To shorten this into a 15-minute talk, this section is going to basically try and cover all the background and theory of capsule networks. And I would love to talk about it for an hour to 2 hours and really fill in all the details. But instead, I'm just going to make a bunch of statements, and you guys are going to convince yourselves that it's true, or you're going to convince yourselves that I'm wrong, and you can email me why I'm wrong. So hopefully you guys will think about it for a little bit if anything doesn't sound right or if anything is a little bit like, is that really true? So why capsules? We can talk about max pooling, affine invariance, and part-whole relationships. I'd really like to thank the speaker who came before me. I don't know if that was the brilliance of the organizers or what it was, but she covered a lot of the things that I had to leave out for time. So keep in mind all the stuff she said, because it directly applies to what I'm presenting right now. So what are capsules? Capsules make two simple changes from a traditional convolutional neural network. Features are now represented as vectors rather than scalar values. And these vectors store orientation information about the input. So when we talk about orientation information, that can be its orientation in Euclidean space. So it's pose, some sort of scale information, some sort of thickness or localized parts or different things like that, which you saw in Hinton's paper. It can also be the orientation in high-dimensional feature space. So if a feature is representing some aspect of the image, its orientation within that space will be the variance of that attribute of the object that we're interested in. The other big thing is the agreement between feature predictions is computed as the weight of the presence and the orientation of high-level features. So this is the dynamic routing part of the algorithm. So basically what we're doing here, so each feature vector makes a prediction for each higher level feature, and then we compute the agreements between the presence of those features and their orientation in space. And we'll see what that looks like in a second. Problem with pooling. So you can convince yourself that pooling is actually a primitive form of routing. And it has some very nice pros, such as the spatial invariance that you gain. And it reduces the memory burden in higher level layers. Right? These are very nice. This is why we use pooling. Pooling is very powerful and has given us some really impressive results in convolutional neural networks. But we mustn't forget the cost that comes with pooling. And that cost is we're throwing away information. We're throwing away information about the input, which we need to do because we're going from a very high entropy input to a very low entropy output. We need to reduce information. Max pooling does it in a somewhat unintelligent way. It doesn't take into account the importance of the features, the homogeneity or the heterogeneity of the region. And capsules are an attempt to sort of fix that issue, where we're using now strided overlapping convolutions and the dynamic routing algorithm to do basically a more intelligent way of reducing information. So we'll still reduce the memory, we'll still get some spatial invariance, but now we're going to actually take into account aspects of the regions when we do downsampling. The affine invariance, so this was covered a little bit in the last talk, but so CNNs are have built-in translation and variance, and that's just as a product of sliding the convolutional filters across the image. The issue with this is that they can't handle the other dimensions of affine transformations. So you can see here, as we take this object and we rotate it into different unique ways, CNNs are not able to handle this. You can convince yourself of that. I'm going to keep moving along. Solutions. So what we can do is we can either replicate our feature detectors on a grid, and that'll grow exponentially with the number of dimensions. Or we can increase the labeled training set to include all of the different possible orientations of the objects, which would not be covered in a standard data augmentation type approach. These are both very costly, exponentially costly in terms of either labels or in terms of computational memory. And capsules are a nice solution to this, which I'll talk about. So the part-whole relationships, this is the most exciting part for me. This is why I really believe capsules are going to be talked about a lot over the next couple of years. But this idea of preserving part-whole relationships. So you can imagine this is some sort of subset of an input space. We send them through a CNN, and we want to do basic classification, right? Is this a cat, a dog, a person, a building, a car, etc.? Your mid-level neurons might be picking out things like eyes, noses, mouths, etc. And your high-level features may be looking for faces. And we saw in several works that have come out where you see examples of these, right, where neurons within a CNN are giving extremely high responses whenever a face is present at that localized region, and they are being fed in from sort of mid-tier neurons that are eyes, noses, mouths, things like that, that are scoring very highly if you actually look at the activation maps of the CNN. If we take these feature maps, I just stretched them out here, what a CNN does is it just takes the sum of the responses of those, and that gives you a single scalar value, whether or not the feature is present at that location. That's why we call it a feature map. And the issue with this is, if the features are present, regardless of their spatial relationship, then this will give us a high value. So if the nose is there, if an eye is there, if the mouth is there, the face is there. Regardless of their orientation, regardless of what kind of face, it's going to give you a high value. That's how CNNs work. If we want to move on to something a little bit more complex and be able to actually describe our data in a slightly more meaningful way and a little bit closer to how our brain actually does it, we need to start talking about capturing orientation information of those features. So you can see an example here, capsule for eyes, capsule for nose, capsule for mouth. What convolutional neural networks do is, so if we take, for example, the capsule for the eyes, we'll project that vector into all of the higher dimensional capsules that we're looking for. So if we have a capsule for dogs' faces, for cats' faces, for humans' faces, it'll say, given this eye, if I was a dog's face, I would look like this. Given this eye, if I was a cat's face, I'd look like this. And given this eye, if I was a human's face, I'd look vaguely like this. Okay? It does the same thing with the noses. And it says if I was a dog's face and I had this kind of nose, this is what I would look like for the cat, for the person. And then same thing for the mouth, right? This is what I would look like. And the dynamic routing algorithm says, well, don't just take the sum of these activations. Let's actually see if they agree with each other, right? And so we run the dynamic routing algorithm and we see all of the dog face predictions from the lower level features are all completely in agreement with each other that yes, this is in fact a dog's face. Whereas the cat face and the human face, all of the features were present. There was a nose, there was an eye, there was a mouth, but they're not at all in agreement about what the face should look like when we project those feature vectors up to the higher level. Hopefully that makes sense to everybody. There it goes. Okay, so yeah, so capsules predict their vectors, voting of the orientation of the features, and then the dynamic routing is what's able to sort of preserve these part-whole relationships and accounts for the other dimensions of the affine transformations. I'll give one more sense on that. So remember, this is not just a scalar value of is there an eye there. It is a vector, which the magnitude of that vector represents whether or not there is an eye there. But the dimensions of that vector tell you about the scale, the orientation, the coloring, all different kinds of aspects of what this eye is. And so we're able to say, given its orientation and its scale, if I was a dog, I can start to talk about what that next higher level feature would look like. Cool. Hopefully that makes sense. So you just hopefully now understand the entire theory of how capsule networks work. If that's not the case, I would highly encourage you to go watch one of the talks on capsule networks. There's some beautiful ones out there. But that should be enough relevant background. So the novelties of the proposed method. So we have locally constrained dynamic routing, transformation matrix sharing, deconvolutional capsules. We're extending the reconstruction to semantic segmentation. And we have a large image data set, and we reduce the number of parameters. So I'll go through these. So the locally constrained dynamic routing. This is the key line here from the paper. And basically what we're doing here is we're saying for each child capsule centered at some position, we're going to project all of those up into all the parent level capsules, and then we'll do the dynamic routing only for each child capsule type within that local region. So we're locally constraining the routing so that we're not routing every single child capsule to every single parent capsule, as was done in the original proposed work. Transformation matrix sharing. This is a quote from the paper by Saburit, Frost, and Hinton. And they're talking about how they're using different transformation matrices for each member of the grid, as well as for each types of capsule. And we changed this to propose that we share the transformation matrices across members of the grid. And this helps us to reduce the number of parameters. There's more details about that in the paper. I encourage you to go check it out. We introduced deconvolutional capsules. This is kind of exactly what you think it would be. We're using the prediction vectors are now formed via transposed convolutions. So we're able to upsample our images. And the routing is computed for each capsule type at a given location. Right? So the routing still stays basically the same. It's just how we're forming the prediction vectors. Reconstruction regularization. So here we extended this to only reconstruct the positive input class of the image, which is what we're basically trying to segment. And at training, we're using the mask. At testing, we're using the vector links. And then there's a potential theoretical explanation I have of why this works and what it's doing. And it relates to the idea of mode collapse in GANs. If anyone's interested in that, I'd love to talk to them about my theories on that and how that's working to represent the distribution of the input data better. This was the first time we had capsules work on large images in a large data set, which was very nice. Typically it was SciFAR 10, 32 by 32 images, MNIST 28 by 28 images. Because of this, a lot of people were sort of calling that, hey, capsules, it sounds really cool in theory, but it doesn't actually work on any real problems. So until somebody figures that out, we're just going to kind of hold off on the whole capsule thing. What was nice is we were actually able to, because we're constraining the routing, because we're sharing the transformation matrices, we're reducing the memory burdens, we're actually able to now deal with 512 by 512 3D medical data on a much larger data set. We're actually able to start applying this to real problems. This is a somewhat preliminary work. We actually we're doing it on all sorts of other medical segmentation tasks, brain, kidney, liver, and some other things. And we also have, applying this to semantic segmentation and computer vision as well. We reduced the number of parameters. So we have 95% fewer parameters than the proposed UNet, and we have 38% fewer parameters than a dense fully convolutional network, Tiramisu. We apply this to pathological lung segmentation. This is a medical conference, right? So we're using the LUNA subset of the LIDC data set, which contains 880 CT scans. There's a lot of difficulties with this data. We have one network architecture, which is very similar to the original Hinton proposed, Saburit proposed architecture. And this is our SegCaps architecture. We followed an encoder-decoder sort of UNet style. We investigated different routing schemes. And And you can see here some qualitative results where UNet is looking at a little bit of over-segmentation or similar HU units. Our network is able to not fall into those same issues. These are the results. So we performed basically on par with all the other results while greatly reducing the parameter burden. We reconstructed the capsule vectors. You can see that the different vectors are looking at different aspects of the input data. And that's it. Yeah, that's the end. So I believe I'm right at my 15 minutes. So.
Yeah, thank you very much for the nice talk. And I guess there are many questions. Yeah, please.
Sure. And while they're running the mic over, I encourage you, any questions that aren't answered, especially people online, the project page, you can comment, and you can send me an email, and there's a link here, and the code's publicly available.
So thanks a lot for a very inspiring talk. I have a question. You say that the parameter size reduces drastically. Does also the computational time reduce so drastically?
No, so the computational time, because of the dynamic routing algorithm, it does go up a little bit. At training, it's about 3 times as slow as UNet, which is, UNet's very quick to train. But it converges usually much quicker. About a half to 2/3 of the total number of epochs are required for convergence. Okay. So a little bit slower.
Thank you very much.
Yeah, further questions? There's no one yet. I can just. Oh, yeah,
sure.
George.
I think this young lady, right
microphone
here.
is running. It's just a quick. So you only showed it on 2D data.
Yes.
You said you want to get it to real world. So well, real world in medical is 3D data.
Yes.
So what's holding off to use it on 3D data?
Partially time. So this is a preliminary work. As I said, we're have results coming right now on all different kinds of organs, moving it to 3D, all the sort of engineering stuff on top of it too, with Atrose convolutions, separable convolutions, all that stuff. So there should hopefully be a bunch of works coming out pretty soon covering.
Okay, but there's no theoretical limit.
There's no theoretical.
In
fact, in 3D it should work much better because pose information in 3D is more meaningful than in 2D. So when we move to 3D, it should hopefully work even better from a theoretical point of view.
Yeah, thank you. Yeah, okay. So a question over there.
Right here.
It's me.
Yeah.
I think
okay.
if there's nobody else. Yeah, I was wondering, because you locally constrain the dynamic routing, does that have any impact on actually the thing that captures are good at with the pose and the rotation?
I do not believe it hinders the pose and the orientation too much. We adopted the encoder-decoder style network to kind of help compensate for that as well. The one thing that I really want to solve that I'm working on right now is capsules are really great. So the eyes, the noses, the mouth, or any eye of any animal in any orientation, capsules should be able to describe that with its feature vector. But because we're now locally constraining each XY, we don't just have one eye, we have a grid of eyes. And in theory, we shouldn't have to do that. In theory, we should only need one eye neuron. And that information be captured of where it is within the vectors. So that'll be another step forward to reducing the memory. But yeah, but I'm not super concerned about your question.
Cool.
There was another question over there.
Yeah.
Yep, George.
On the back. And while I was running there, again, anyone has any other questions, please just email me or comment on the project page. This was a very quick run through of a pretty dense work, especially if you read the paper. It's a ton of math all over the place.
So very quickly, in terms of human anatomy, obviously the relationships between organs and the orientations between pathological features in images and related organs is extremely important. Probably the lungs is one of those areas where that's least important because, I mean, when you look at those pathologies, it might not be the most important application. But for example, when you look into, let's say, the brain, and you look into, I don't know, perivascular spaces that have specific orientations relative to other structures, then the orientation between pairs of features that activate or different neurons that activate becomes really important. However, those relationships tend to be of relatively long distance, so sometimes they're a little bit far out. So I guess my question was, have you thought about what will be the application in human anatomy or pathology where relationships between orientations of activations or of objects becomes important, but while they are at the same time spatially constrained to a relatively small field of view because of the rerouting limitation that you have?
Sure. So short answer is yes. The longer answer is using this sort of framework to actually start to perform characterization of pathologies is definitely a direction we're going. In a perfect theoretical world, a generalized framework for imaging biomarker discovery would hopefully be theoretically possible with this. That's the direction we want to go, and we can talk about that offline if you'd like. But in short, yeah, the relationship between different imaging features should tell you some information relevant to the pathologies and relevant to biomarker discovery. Yeah, there's some interesting thoughts there that I'm currently working on.
Yeah, we can catch up.
Yeah, yeah.
Okay, yeah, so let's thank the speaker again.
Thank you.
And